Well, we're very excited today. We have a first timer appearing on the Healthcare Unfiltered podcast. And Tom, I promise if you play your cards right, I'll invite you again. I tell you, I promise that.
Sounds great.
Well, welcome to the show, Dr. Tom Beer, being with us on Healthcare Unfiltered, and we're going to talk about really a lot of exciting stuff, but focusing a bit on multi-cancer early detection testing and things of that nature, because it's really a hot topic. A lot of people are talking about. It's not uncommon that you see a lot of these sessions at scientific meetings and scientific conferences and things like that. But Tom, before we get started, maybe a bit about you and introduce yourself because you hold a very high position at Exact Sciences.
Yeah, thank you very much for having me today. It's really a pleasure to talk with you. A bit about me, I'm a medical oncologist. I take care of men with prostate cancer. I still do to this day. I have had my entire career at the Knight Cancer Institute at Oregon Health and Science University in Portland, Oregon, and I mostly focused on a prostate cancer clinical trials program and patient care program for many, many years. I ended up serving as deputy director of the Cancer Center, and during that time, uh, saw us put together a Center for Early Detection after a large fundraising campaign called the Knight Cancer Challenge, supported by Phil and Penny Knight, who challenged us with a $500 million if we could raise another $500 million. And we succeeded with the help of many, many Oregonians, Washingtonians, and people from all around the country and the world. And with that investment, among other things, focused on building a center for early detection, attracted a wonderful group of scientists and engineers. And after that center got off the ground, it became clear to us that we needed a clinician involved. And as deputy director of the Cancer Center, I felt a responsibility to do that, stepped in as chief medical officer for the center, and really fell in love with early detection. So that was really how I got involved in this field. In that role, I got exposed to all the new technologies for the emerging tests in cancer screening, got a chance to do research with our own scientists, as well as collaborate with many industry partners in this area. And then a couple of years ago, maybe 2.5 years ago now, I heard from Exact. They were looking for a chief medical officer for their multi-cancer program, and I just could not pass up that opportunity. And so I've been there a little over 2 years now, providing medical leadership to our MCED team.
It's a amazing story, actually. But let's go back a bit. What got you into geocancers in the beginning? Was it somebody you met in fellowship? What got you into prostate cancer?
I had a wonderful mentor when I was a fellow, Dave Henner. Dave was a breast medical oncologist. I was working in his lab, and I said to Dave, I'd like a job here at Oregon. What should I do? And he said, well, no one's doing prostate cancer, so why don't I answered that? And it really was like that. And you know how it is. I mean, once you get involved in something, it becomes a big part of your professional life, your professional identity. So, I feel very much a part of the prostate cancer community of patients, families, physicians, and researchers around the US and the world. But the start was really quite humble. I just wanted a job, and that opportunity presented itself, and I jumped at it.
It's amazing how these things go one day. And yeah, we've met a few times back in the day at ASCO GU. That's why, I mean, you're obviously continue to be a phenomenal figure in the GU oncology community. I did not know that story of raising $500 million. I mean, this is really amazing accomplishments. Congratulations. How did he do that? I mean, this is not, I mean, we're not talking few millions. I mean, this is crazy.
Yeah, so that was quite an adventure. Most of the credit for that work goes to Joe Robertson, who was president of the university at that time, and Brian Drucker, who was the cancer center director, now is the chief executive officer of the Knight Cancer Institute. And, a legion of folks from the foundation and around the Cancer Institute helped. But Joe and Brian really did the bulk of the work. And what happened there is, Phil and Penny Knight actually surprised us at a gala with a challenge. I remember the moment. Phil made
Wow.
the announcement that they would put up $500 million if we could raise the other half for a total of $1 billion. And we, at that point, with Brian's leadership, of course, rolled up our sleeves, articulated a vision for investing in a large number of priorities, but the big new priority was cancer early detection, and really got engagement from so many people. The Oregon legislature put up $200 million towards that $500 million challenge. And then countless donors, I think it was more than 10,000 donors,
Wow.
most from the state of Oregon, but from around the country and the world, really resonated with the vision and contributed. So it was a sustained effort. It took, I think, almost 2 years to meet that challenge. But we did. And
Amazing.
the Knight Cancer Institute has really grown tremendously ever since.
Amazing. Yeah. I mean, I've known Brian since the CML world, right? I mean, I did a lot of heme malignancies, so it's hard not to get to know Brian.
He's
Amazing.
been a tremendous fundraiser too.
Yeah. Yeah, you can't be a, cancer center director and a CEO of an entity like this unless you know how to get money. So right now you transitioned into your chief medical officer role at Exact, but you're still seeing patients?
Yeah, so I'm fortunate enough to have an adjunct faculty appointment at the Knight Cancer Institute. I serve as adjunct professor of medicine. In practice, what that means is a half a day of clinic on Friday mornings, where the Friday clinic day is reserved for adjunct faculty by and large.
Well, at least you didn't get the Friday afternoon though. I mean, you
No,
could
you're
have
right.
gotten like the
Yeah,
1 to
you're
5
right.
p.m. on Friday.
You're right. Yes, I'm fortunate to have Friday mornings. It's also the day that Exact Sciences chief medical officers practice. So both institutions have coalesced on Fridays. And it's really wonderful to be in the clinic. I still see the same patients. I've seen some of them for a decade or longer with the same clinical nursing
Yeah.
staff that I've worked with for years. Most of my time, though, I spent serving as chief medical officer for multi-cancer early detection at Exact. And that involves a fair amount of travel to Madison and travel all over the country and sometimes out of the country and occasionally work from my home office.
I was going to ask you, I mean, what does your day look like? How do you spend your day or week in general? I mean, how much of it is, kind of relationship and talking to investigators and researchers versus really the, rolling your sleeves internally and talking to the folks in R&D and things of that nature? How do you split your time?
Yeah. Well, so I think the role of CMO at the most basic level is to represent the patient in the C-suite. That's what a CMO is supposed to do. Um, but in practice, there's several core functions. So there's providing medical leadership to the MCED program, and so that means engaging with the clinical development and the clinical operations teams around clinical trial design and execution, overseeing the trials as they go, making sure that from a medical perspective, everything's on track, helping oversee the monitoring for any adverse events and developments on a trial, working with the regulatory team to uh, prepare interactions with the FDA and making sure that the regulatory oversight function is fully supported from a medical perspective. And then just being a part of the company leadership team and thinking strategically about how we might advance cancer screening and cancer early detection across our portfolio. And then, externally, there's a pretty substantial role in representing the company and its vision and its MCED program to a variety of external stakeholders. The most important there are scientific meetings and our interactions with patients and advocacy organizations. So I get a chance to um, go to ASCO and ESMO and AACR and all the meetings I used to go to when I was on faculty, except the prostate specific meetings.
Yeah,
That's
yeah.
so
So, so, so
can't go
uh,
by those.
at Exact, you have obviously several programs. So you have various CMOs for various programs. So you focus specifically only on the MCED, but there are other folks who are focused on other top programs.
Yeah, my responsibilities for MCED, Exact has grown as a company both organically and through acquisition. And so my original hiring title was a CMO of Thrive. Thrive was the company that launched with technology from Johns Hopkins and started the work on MCED in Baltimore, then in Cambridge and Boston, and was acquired by Exact Sciences before I came on board. The technology from Thrive was then combined with Exact Sciences technology, but I'm essentially CMO for that portion of the company. Now, recently, I've taken on some other additional responsibilities, but within the company, we have a chief medical officer for screening. That's Paul Limburg. He's a gastroenterologist at the Mayo Clinic, and his number one responsibility is overseeing the colorectal screening portfolio, but he also has other screening responsibilities. And then we have a chief medical officer for precision oncology, which used to be Genomic Health before Genomic Health joined Exact Sciences. That's currently Rick Boehner. He's a pathologist at UCSF. You can sort of see the theme. We've got different professional backgrounds, different academic affiliations that kind of make sense. Molecular pathology for precision oncology, gastroenterology for our screening franchise that is really centered on colorectal cancer, and then medical oncology involved in multi-cancer. And we all work very closely together, cover for one another, and there's some overlap between us.
Of course. Do you ever miss being full-time faculty and the grind of hardcore academic medicine?
Of course I do. I mean, I was very fortunate to do what I did, and I miss a lot of aspects to it. I think the opportunity to be a part of the GU oncology and the prostate oncology family, if you will, and it really that community feels like a family is something that I remember very fondly. I still try to show up for those sessions whenever I can and see my friends. So I do miss that. I miss some of the mentoring opportunities that I had at the Knight Cancer Institute. I miss my prostate cancer team, both in the clinic and in the clinical research arena. I miss working with Brian and helping steer the cancer center. Certainly, the Center for Early Detection, where I served as CMO, is just a wonderful organization led by Sadek Esner, and miss being there. But I wouldn't trade my current job for those. I mean,
Yeah.
I had
I mean, there's a
time.
there's
It was
a
wonderful.
time for
Um,
everything.
there was things I miss, and I will always remember fondly.
Yeah.
But the chance to be chief medical officer for MCED at Exact and to work hard to try to really transform cancer screening is not something that I would trade for
Yeah.
anything else, really.
Absolutely. But it's funny when you mention about the GU community. It has just exploded with newer therapies, newer targets. Like really, I mean, urothelial cancer. I mean, it's just like it's nuts. I've done so many podcasts on this, and it's like I feel every time I say, okay, I'm okay, I can like wait maybe 6 months before do another one. I can't. Like there's a lot of things happening. I mean, so really, GU oncology have transformed a lot since I started seeing patients over 20 years ago. So kudos to all of the scientists.
Yeah, there's been tremendous progress across oncology, and which nowadays I think about a bit because I think when you think in a different lens. So when you think about cancer
Hmm.
screening and early detection, of course, all the human benefit of cancer screening comes from the detection and the intervention that follows, right? So as interventions get better, um, impact of cancer screening tests is going to get better. even if we don't make the tests any better themselves, which of course we're working hard to do,
Yeah.
the fact that the interventions are getting more effective is really compelling for the not just for the medical oncology treatment side of the equation, but also for those of us who are working on cancer early detection.
So we're going to talk about multi-cancer early detection a little bit, but I wanted to put the hat that this is not everybody who is listening is, of your knowledge base. There are various different people that listen to this show. Some of them could be patients, families, others could be oncologists or students, fellows, residents. So multi-cancer early detection, simplify that concept to the lay people who may be listening to this show.
Absolutely. So let me start with the why we are pursuing this, and then I'll talk about how it is structured. So with cancer screening today, we know that detecting cancers earlier can lead to better outcomes. And in several cancers, we have definitive evidence from large studies that cancer screening can uh, reduce cancer mortality and save lives. So we think this is fundamentally a good idea to pursue. But today, we only have 4 or 5 cancers for which routine screening tests are available for the general population. Depends how you count them, but I think we all agree on cervical cancer screening, colorectal cancer screening, breast cancer screening. Lung cancer screening is available for smokers, but for those folks who aren't smokers or haven't smoked more than 20 pack years, we don't have routine screening available. And then PSA screening is available for prostate cancer and has been subject to some debate and is available as a grade C recommendation from the USPSTF. That covers about a third of incident cancers and cancer deaths, depending kind of how you count it. So the majority of cancer cases or cancer diagnoses and the majority of cancer deaths occur due to all the other cancers for which we don't routinely screen for today. That's the primary objective for multi-cancer early detection or MCED tests. The idea there is that we will use our understanding of cancer biology to measure in the bloodstream biomarkers or substances that are detectable in the blood that come from a variety of cancers. And if we're successful, hopefully design a single blood test that could detect multiple different cancer types and expand the reach of cancer screening to a much larger proportion of the cancer burden that we can screen for today. That's the vision. Now, this is a field that's in development. So this is currently, I think, a vision that we're working to realize.
But But Tom, like you're a GU oncologist, and you mentioned prostate cancer and its grade C and the PSA, and you mentioned other tests. I mean, it's one of the challenges is that you can't really tell by screening how much you're really saving lives. It could be there's like you have an early diagnosis, which could lead like this lead time bias type of thing when, maybe I'm detecting something especially in prostate. I know you'll appreciate that obviously more with prostate because you could detect something, but it may not be really clinically significant. It may not mean much. When you're doing these things with the blood, and I'm sure you will get to it, but are you factoring that possibility that I may be detecting something, but it's almost like, okay, it's there, but it's not going to cause any trouble.
Yeah, no, we're thinking very hard about that. And screening for cancer, when you really think about it, is incredibly complex and interesting. So, what you alluded to there in your question is the problem of over detection, over diagnosis, the idea that a screening test might pick up what we might call a clinically insignificant cancer. Now, for those of us in the audience here who are not aficionados of cancer screening, I always get a question: what's a clinically insignificant cancer? Isn't all cancer important? And the answer is that a clinically insignificant cancer is typically a small, slow-growing cancer that might not cause any harm if left untreated for the remaining natural lifespan of that person. And that those kinds of cancers do exist, and we need to be very mindful of those because detecting those cancers early might actually not help folks and might actually be net harmful if we start treating people unnecessarily. And this is a challenge that we encountered in the prostate cancer field. Prostate cancers tend to be very slow growing, and some of them are very aggressive, some of them are very slow growing. It's a real mix, and we have this paradoxical situation where prostate cancer, this is the second leading cancer killer of men in the United States. And yet many prostate cancers are slow and clinically insignificant. And that's challenged the field. When PSA screening came along early on, it was difficult to tell the difference. And a lot of men got treated for cancers that probably didn't need to be treated. Now, the field learned from this. And so we now have a well-established approach called active surveillance, and really a much better understanding of which cancers are indolent or slow, which cancers are aggressive or fast. We can use histology, the so-called Gleason score. We can use molecular analyses of prostate cancer to predict the behavior of the disease. And if we believe it's one of those slow-moving cancers, what we typically recommend is observation or active surveillance and not immediate treatment. So the field of prostate cancer has not stood still. It's responded to this opportunity and challenge in a way to try to capture the benefits of screening while minimizing the harms. But the story is different cancer by cancer. So for example, in colorectal cancer, we want to detect not just early cancers, but precancer, because there a colonoscopy can then be done, and these precancers, these polyps can be removed, and we believe that actually can reduce the incidence of colorectal cancer and colorectal cancer mortality. And when modeling analyses are done of colorectal cancer screening, it turns out that precancer detection is the major contributor to the mortality benefits of colorectal cancer screening. So there's a lot of subtlety and complexity. In some cancers, overdiagnosis is a real concern. In other cancers, it's much less of a concern. And so we really have to think about this problem on a cancer by cancer basis.
And I'll dig into that a bit deeper in a bit, but let's just go back to the basics. You said we do a blood test. Like, what are you checking in the blood? Just what are you taking the blood tubes, and what are you actually checking?
So there's a variety of approaches to that. At Exact Sciences, we're currently looking at a series of protein biomarkers and a series of DNA methylation biomarkers. So what we detect in the blood there is the DNA that comes from the cancer. DNA, of course, is the nucleic acid that codes for human life. And DNA circulates in the blood, both from cancer and from normal human tissue. We're able to look for methylation patterns that are unique to cancer, and that's how we use DNA to look for cancer in the blood. Methylation is a chemical modification of DNA that happens both in normal tissues and in cancer, but it happens in different locations depending on whether we're looking at a cancer or a normal cell DNA. There's also DNA mutations, and one of the prior versions of our assay looked at DNA mutations, and we're continuing to evaluate DNA mutations as a biomarker for potential inclusion in our assay. And there's other things you can look for. You can look for extracellular vesicles. You can look for aneuploidy. You can look for DNA fragmentation. So different companies that are developing tests in this space are looking at different biomarkers. But what they all share in common is that these are blood-based biomarkers. They're released from cancer. And very interestingly, they're often shared by many different cancers. So the proteins and the DNA methylation sites and the DNA mutations that we and others are looking for come frequently from different types of cancers, which is why a single blood test is capable of detecting a variety of cancer types.
I mean, are there cancers that maybe they are not going to, you know, shed things into the blood that you may be able to detect? I mean, is there a possibility that somebody could have a cancer that obviously we don't see it visibly yet, but somehow these cancer cells are not really secreting whatever you're trying to detect in the blood? Or if there is cancer, you'll be able to detect it in the blood. I guess what I'm trying to get at, Tom, is the sensitivity and the specificity of what you're trying to get, and how, A, are there cancers that may not shed any of these molecules or fragmentation that you're trying to look at? And B, there's no assay that will ever be 100% sensitive and 100% specific. So there are trade-offs. How do you really design something where you can be more comfortable with the output?
Yeah, so a lot to unpack there. But first of all, there are differences between cancers. So although the biomarkers are shared across a variety of cancers, as you alluded to, the shedding levels differ. And so you can cite examples such as thyroid, breast, and prostate cancer as an example. These are relatively lower shedding cancers. And so the sensitivity of these multi-cancer tests, whether ours or others, tends to be lower for those cancers. And that's not an exhaustive list. Those are just some examples. On the other end of the spectrum, pancreatic cancer, lung cancer, ovarian cancer tend to be relatively high shedders. And we've presented at a recent meeting in ESMO our data on sensitivity by cancer types. So have others. And so people are free to take a look at that. And you can see that there is a range of sensitivities. One of the things that we're observing in the field that's interesting is that there does appear to be a correlation between tumor aggressiveness and the level of shedding. So this problem of over-detection, of detecting insignificant, small, slow-growing cancers may be less of an issue for this class of tests than it was for the PSA test for prostate cancer, for example. We'll see. But for now, it does look like the tests tend to detect the more aggressive cancers. The sensitivity, even for the high shedding cancers, is not as high as we would like it to be. So these tests will, without a doubt, not pick up every last cancer. What that sensitivity will end up being, time will tell. We need prospective studies to determine that. In case control studies, these are studies where we evaluate blood specimens from people with known cancers and also people who are thought to be healthy. The overall sensitivity across all stages is around 50%. But it is higher for high stages and lower for early stages, which is what we would like to maximize sensitivity. We expect that when we take that type of test into the screening population, that sensitivity will be lower. We don't know how much lower, time will tell, but case control studies are not the same as
Yeah.
putting that test into practice in the what we affectionately called intended use setting, or the actual human seeking cancer.
I want to pivot in a bit to the Falcon trial, but before I do that, just in general, Tom, like conceptually, whenever we think of screening, we're always thinking of a patient population at the highest risk of developing a particular disease. Even in prostate cancer, despite how lousy the PSA is, obviously, I mean, it's not awful, but it's not great. You don't really do PSA for a 25-year-old healthy man. Breast cancer, the same, even colorectal cancer, things like that. What concerns me about the MCED is that I see a lot of folks saying, well, let's use the MCED for everybody. Do you think we should do MCED and test MCED in people who are at highest risk of developing the disease, or do you think MCED should be done in anyone who wants to check? Like what general population or select population, I guess?
Well, I think it's too early to take an informed position. So I will just share with you what my thinking has been about this. So I think there's no question that the test will have certain performance characteristics, sensitivity and specificity. We and others are designing these tests to have a high specificity to limit false positives. So they are fairly high specificity, and the trade-off there is somewhat lower sensitivity. But you have to have a certain amount of pre-existing cancer risk for that balance between false positives and true positives to make clinical sense. So I completely agree with your assertion that it would be quite difficult to conceive of a test like this for truly everybody, starting at age 18 or something like that. And I get this question, and it's a heartbreaking question sometimes that I get from folks who've dealt with pediatric cancers, parents whose kids have had pediatric cancers, and they ask, "What about us?" And it's hard to explain, at an emotional level. It's hard to answer that question and say, "I hear the pain you went through, but the math doesn't work very well. Kids' cancers are uncommon, and we would cause a lot more false positives than it would make clinical sense."
Yeah.
So that is a tough discussion with people who've been touched by cancer in their family and are seeking answers for how to protect themselves and others. Right now, the way we've designed the program is age 50 and up. We will be looking at risk groups. So that's where we've designed our program, our study, and we'll be looking at the outcomes of our studies by age group, by smoking status, by family history, and other indicators of risk to try to best understand performance in each category. And it would be premature for me today to make a definitive recommendation, but no question we're thinking about how to balance the desire of average folks to have access to cancer screening with focusing on populations where the need is greatest.
I was intrigued when I read the announcement that you had in the press on the Falcon trial because it's a huge effort from just what I read, for whatever in the press release that you had, and I'm going to have you tell folks what it is. But I mean, 25,000 patients, if I recall. I mean that's a lot of investment in resources and effort to really go in and just see what you get the results, which we don't know what they are, but it seems that you really wanted to at least study this comprehensively. Tell us what is the Falcon trial and what led to the inception of the trial?
Sure, absolutely. Thanks for that question. So, yeah, I mean, 25,000 patients,
Or
turns
people, they're not, we don't
out,
know if they're patients.
right, 25,000 individuals. That turns out to be an intermediate sized trial in the screening space. So
Yeah.
that kind of drives home the point of how big a task it is to really evaluate these tests properly. What we're doing in Falcon is we are enrolling up to 25,000. We'll obviously look at our lessons as we go, and the goal is really to evaluate all aspects of an MCED test in a real world setting. It's a real world evidence gathering study. It is carried out with two large health system partners, Baylor Scott and White and Endeavor Health. It's up and running at Baylor Scott and White at this point, and Endeavor Health will be activating here later this fall. And we will be looking at really everything from the ability to reach a diverse population of patients to offer the test to how patients decide to go ahead to what the diagnostic experience will be, how efficient our workup strategy proves to be in the real world, how the test delivers on its performance. And we'll be using all those lessons to really refine our approach, learn, and ultimately to inform the design of our pivotal trial that would be designed to seek FDA approval for the test. So this is our way of really getting getting the learnings that we need to develop this test responsibly going forward.
One of the most challenging things that I was thinking about this when I read the your release on the Falcon is that it's very easy to decide when the test is negative because you could say maybe we'll do it again in a couple of years. I don't know the design, but you'll just repeat it in a year or 2 years, however the design of the study. I think what's challenging in my mind is when the test is positive and clearly you have to do imaging after that. But how often, let's say you do imaging and you find nothing. Let's do you do CT and I don't know if there's a PET or not. You do a CT or a PET or whatever it is and you find nothing. How do you determine how often you need to repeat the imaging study? Because it's empiric at best. And you could say 3 months, you could say 6 months. I mean, I don't know. How do you design this in a way that does not appear arbitrary, although in my simple mind, it seems like it has to be empiric because there's no science behind how frequent you're going to repeat the imaging study when it's positive.
Yeah, we don't have a definitive answer to that question right now, but I will say that this is a challenge that all screening tests have. So we have false positive results for every kind of screening test. In fact, our conventional screening tests of today will have false positive rates of 10% or so, 10, 11, 12%. With the MCED as designed, it's about 1%. So about 1 out of 100 individuals might expect to get a positive result, but a negative workup.
That's
I
that's
think,
not bad. That's pretty good. Only 1%?
yeah, that's the high specificity piece. We're really
Right, right.
trying
I see. I see. I
to
see. Yeah, yeah.
limit that. So, um, with conventional screening tests, we've gained the experience to know that if you get a negative colonoscopy after a stool-based test, you can go back to routine screening. We've got lots of data to support that, and people are comfortable with that. Um, we've looked at all the data that we have that can inform this question, and that was the DETECT-A study, where a prototype of our test was evaluated in 10,000 women, 10,006 to be precise. That's a pilot feasibility study in the screening space. So again, you can see how big an
Wow.
effort is required. There were 98 individuals who had a false positive result, meaning they had a positive test result and no cancer was found on an evaluation that involved both CT imaging and a PET scan. We now have about 4.5 years of data on those individuals, and their risk of cancer was no higher than what you'd expect in women aged 65 to 75. It was about 0.7% per year. There were 3 cancer cases diagnosed in the ensuing 4.5 years. So that gives us preliminary confidence that the diagnostic workup that we've designed, if it's negative, that individual really should be able to go back to their routine care and not walk around thinking that they're at elevated risk, that there's a hidden cancer somewhere that was missed. That's the outcome I hope to confirm in the Falcon study. But the preliminary data so far, basically, I mean, you could summarize it with, if you have a negative PET scan, that's really good news. And that's what it appears to be. In the Falcon study, what we do is we recommend that folks go back to all of their routine normal cancer screenings that would be recommended, and they get a repeat MCED test in a year. We're not scheduling intensive imaging in those folks. We don't currently have evidence to suggest that that should be necessary. We are monitoring closely. We're going to see what happens. And if there's a signal that suggests otherwise, obviously we'll run that to the ground and figure it out.
So if the test is positive, you're going to repeat the test in a year. You'll do baseline imaging, and then you tell the person just to do their routine screening.
Based on the data we have right now, if we do a thorough diagnostic workup with a clinician evaluation, basic labs, a CT scan followed by a PET scan if the CT scan's not informative, the data that we have suggests that that individual is not at elevated risk of cancer and should safely be able to go back to routine screening. So that's the current approach. again, early. We're studying it. We're going to continue to learn. And it's not a recommendation for routine
Of
practice. It's
course, of course,
how
of
we've
course.
approached our current study.
Yeah. And with up to 25,000, that may take a couple of years, I presume, until you get something. And then after that, you're going to run the confirmatory trial based on the study that you're like. What's the end point of Falcon? Is just more like feasibility and exploratory stuff, or there's a hardcore endpoint?
It's a real world evidence study, so it's designed to collect data and allow us to analyze a broad range of endpoints, but it's not powered around performance or something like that. It's really a broad learnings type of study. It's an RWE protocol, so it doesn't have the classic
Yeah,
sort of
yeah.
primary secondary endpoints in its design. We'll be looking at everything, and we will be following patients on Falcon for 5 years at a minimum. We'll be offering repeat testing on 3 occasions, so after 1 year and after 2 years. Uh, we're not sure, by the way, that annual is the right interval. That's what we hope to learn. People have assumed that you repeat it every year. We'll see. It may be sufficient to repeat these tests every other year. That would be much more economical and expose patients to fewer false positives. So we're open to exploring a variety of intervals. Cologuard is done every 3 years, and that's where we feel the optimal balance is between risk and benefit and the greatest benefit
Do you need
captured.
to like randomize patients, you think, to like various frequent, it's be very tough randomizing to 1 year, 2 years, 3 years, or do you think like this will
I,
be more
I
retrospective?
think I think in our case, we're going to learn what we learn with the second test, and
Yeah.
and we'll think about that deeply and build in an optimal
Yeah.
schedule into the pivotal trial. The pivotal trial, as we've envisioned it, and it's not up and running at this point, is a randomized study where people get randomly assigned to standard of care screening, routine care, or standard of care screening plus an MCED, and that would enable us then to compare outcomes and the impact of MCED. So that's the ultimate vision for how we prove out the utility of this test.
Well, and congrats for thinking through this from the randomized control trial lens, because that's frankly going to be more definitive in answering this question more than speculative. So that's really very important. Maybe just my last couple of questions, Tom, and you've been very generous with your time. One is, what are your thoughts about MCEDs? Like, do you think MCEDs should be ordered only by a healthcare provider, or do you think as they become more available, I don't know, 5 years from now, I mean, like now, 23andMe, right? I mean, you could go and just order 23andMe. I don't need you to write me an order to get 23andMe. Do you think we're heading this way where I don't need you to tell me do an MCED, I just go and get my MCED done?
Boy, for the foreseeable future, it's hard for me to imagine just a purely consumer-driven order. Cancer screening tests remain really the providence of clinicians, and there's a role for an informed consent process to understand the risks and benefits of screening. Like any other medical procedure, as we alluded to in several of the questions we discussed, there are risks. There could be a false positive result that might have implications for that person. So I'm not at this point imagining just cutting the healthcare provider out of the system altogether. Having said that, what we do see is annual checkups, preventive medicine style visits with primary care providers are going down in the United States. It's hard to get an appointment. Access to those kinds of services, unfortunately, is not universal and uniformly distributed across our population. So, we're going to have to think about in innovative ways if these tests move forward, which we will need to prove them out first, of course, how we make them accessible and how we bring them to everyone in the country so that access is universal and equal. And so I think we'll have to be innovative about it, but I don't see a 23andMe style approach where no clinician is involved. We really need that clinician to help patients understand the risks and benefits of testing. We would like MCED tests to be part of a broad cancer reduction program, that folks don't take these tests and forget to get their colon cancer screening or their mammography, but instead get these tests in the context of an encounter where they get reminded that this is part of a puzzle, and maybe we can leverage the MCED interaction to increase and not diminish adherence with proven cancer screening. We need clinicians in the loop to help evaluate a positive result and advise patients what to do. So I think it's hard for me to imagine a clinician-free MCED care
Yeah.
pathway.
Yeah. Yeah, I tend to agree with that, and I think will be interesting to watch this field that it evolves over the next few years. So if I have you back in, let's say, 3 years from now, where do you think where do you think we'll be? Are you able to put your futuristic hat on and see where that field is evolving? It's probably one of the most a lot of people are excited about it, and we always wonder where do you think we're heading?
Yeah, no, I hope to be back even sooner than 3 years,
Oh,
but
we'll bring you back for something else, but for
with that shameless plug, let me try to answer your question. So, I think the field is very dynamic, and there's a lot of investment, a lot of activity. 3 years from now, I would certainly hope to have results from Falcon, at least from the first year or two, that would inform our program. I think other folks are moving a lot of research forward. Having said that, because this is cancer screening, um, developing really definitive evidence of safety, efficacy, and clinical utility, which are sort of the three pillars in the field, takes a long time. And
yeah.
so I think 3 years from now, we're still going to be knee deep in research and working hard to develop and advance these tests towards the clinic. We will, there may be some limited availability of these tests clinically, but I don't see the evidence base for widespread adoption, at least for our test, being available in 3 years. I think you'll see some adoption of these tests from various companies, but more work will need to be done even 3 years from now.
Well, Tom, this was great. This was really nice to give like an overview on MCED, the positives, negatives, and what you're doing with the Falcon trial and additional studies. Anything I should have asked you that I completely overlooked and I forgot?
Well, there's a whole discussion one could have about what kinds of clinical study endpoints one would rely on to get comfortable recommending these tests to the public. And there's a rich debate on that between relying on mortality, which would require probably 15 years and an enormous investment, and seeking some evidence that can be acquired earlier and get us moving towards clinical implementation in a world where 600,000 Americans and 10 million people worldwide are dying from cancer. So that's a rich debate. It's probably a discussion for a whole nother
That
program if you
is
have
that is
time.
definitely actually that is a really interesting point, and I agree. I did not ask you. This is a completely different podcast. That maybe it's a nice debate to bring several folks to discuss this. I'll have to say, I mean, no matter any way you look at it, though, I mean, the Holy Grail will remain: are people living longer or better? I mean, at the end of the day, the screening debate always. Are we
Yeah.
really making, are we reducing overall mortality or cancer specific mortality? And I think for the pure academician in us, I mean, overall mortality, because that's the only way to factor in the possibility of other confounding factors that might really affect things. But it's a fair question. It's totally a fair question.
Yeah, I think it'd be a wonderful discussion. And you said living longer or better, and I think both of those are, for a variety of medical interventions, endpoints of clinical benefit. And the question is, I think no one would disagree that reducing cancer mortality is the primary goal of screening. Overall is tough. That's a long discussion, but overall mortality integrates everything that can happen to people, and it's a tall order to ask a cancer screening test to impact on overall mortality. But cancer-specific mortality is a pretty common endpoint. To me, the question is, can we figure that out with enough certainty earlier, or do we really need to wait until people die? And that's the puzzle. I don't have an easy answer to that, but I can tell you that there's a lot of interesting thought and creative ideas out there for how we might be able to detect signals that enable us to predict mortality benefits sooner than when you and I are both retired.
Well, I this is definitely a debate that you got me intrigued in. I need to think. Well, you're definitely going to be part of it. I just have to think who's the other person now. Dr. Tom Beer, Chief Medical Officer of the MCED program at Exact Sciences. Thank you so much for coming on Healthcare Unfiltered.
It's been a real pleasure. Thank you for having me on the show.